more and more individuals with a wide variety of epilepsy or associated neurological conditions are starting to have genetic testing. And this means that we can get a much better idea of, one, who are the types of individuals and what are their clinical features who have mutations in CHD2, but also do they fall within specific parts of the CHD2 protein. So the CHD2 gene encodes for a CHD2 protein. It's really large. And so there are a number of different changes within the gene that can cause epilepsy. But there are changes within this gene that are also present in the general population and don't cause epilepsy. And so it's really useful for us to figure out which part of the protein is most key in terms of causing epilepsy. Trying to find the cause of these types of epilepsies helps us think about targeted treatments. Just having a big black box of this individual has epilepsy and doesn't respond to a lot of the anti-seizure medications that we have is not terribly helpful, right? But the first step is really identifying that gene. CHD2 is a gene called a chromatin remodeler. And that sounds fancy, but really the job of this gene, or the protein that it codes for, is in the cell. It is responsible for turning other genes on or off. And what I mean by that is when a gene is on, it basically means it's making protein, and when it's off, it means it's not. And so we know that is CHD2's role in the cell. What we really don't have a good feel for is what are the genes that it's turning on and off during the development and the functioning of the brain. And so that's really one of the key questions that we're trying to answer in the lab. And so what we do is we use stem cell models. So these are typically cells that we have reprogrammed to be stem cells. And the original source is a blood sample or a skin punch biopsy from an individual who has a CHD2 mutation. And we reprogram these into stem cells, and then they can be differentiated into neurons in a dish or even organoids. And so we use these models to figure out which of the genes that CHD2 is turning on or off, but also how it does that. And so our hope is once we've identified those, as well as associated phenotypes, so for instance, do the organoids grow a little slower? Do they take on slightly different properties? Having those readouts or phenotypes then allows us to start testing therapeutics. And so in terms of the neurons and the organoids that we're working with, where are we? I would say that those experiments, it's pretty early days. I would say that we're probably 2 or 3 years from kind of having a good understanding of what CHD2 does in the developing brain. In terms of therapeutics, there are so many companies now that are testing different therapeutic modalities that I think tailoring them to CHD2 won't be difficult. The big bottleneck right now is having a good model to test them in. These models, is what we call them, are really the first step towards targeted therapeutics. If we don't know what happens when you mutate CHD2, even if we could design the best therapeutic in the world, if we don't have the models to be able to tell if it works, then it's pretty much useless. These models take months and months to establish, just getting the stem cells. So that can take 3 to 4 months. Once we have those stem cells, we then need to differentiate them into neurons or into those organoids, so those 3D structures that model the developing brain. And that takes, before we can even begin to do studies on those models, it takes anywhere between 2 and 6 months to really even be able to begin our experiments. And so that's hugely challenging and hugely frustrating for families. I think the other part of that is keeping those organoids and those neurons alive for 2 months is super challenging and super expensive. It also means that all of the fantastic people in my lab are coming in on weekends and feeding the little cells and keeping them happy. So I think stem cell models are hugely important in terms of driving targeted therapies, but I think they are a challenge and they are expensive. Is it 5 years away? Is it 10 years away? I don't know. I think it'll vary from condition to condition. Some of the genes that we first discovered in epilepsy, so one I can think of at the top of my head is a gene called SCM1A. It was discovered about 10 years ago. And if you'd asked me if there would be a targeted therapy, so a therapy that specifically addresses correcting the SCM1A mutation, I would have said no way. But now there are 2-3 clinical trials that are ongoing that try and address the lack of SCM1A in individuals with epilepsies. And so I think it's moving rapidly. While CHD2 research might be a little bit behind, and again, that's just a product of the gene being identified more recently and us not having a good understanding of the biology. Even though we're a little bit behind, we can learn from what other groups have done. What types of treatments work, which ones don't, which types of models are the best ones to be using. So are stem cells model more beneficial in one scenario versus a mouse model in another scenario? We'll begin to learn from the other groups. And again, because there's such a close sense of community, all of the family foundations work together, the overwhelming majority of the researchers work together, and I really feel that as a field, we'll be moving forward and learning from other groups as well. And once we find those phenotypes, we'll be ready to jump and test the therapies in our models, but also zebrafish models, mouse models that other colleagues are working on.